Good afternoon, everybody. Thank you so much for joining us today for our speaker series. My name is Stephanie. I will be moderating things here on the back end. So when you have questions, you can put them in the Q&A or the chat, and I will relay them to Dr. Heidebrink. I will also be sending out a Qualtrics evaluation in the chat towards the end of this presentation. And without further ado, I'd like to introduce Dr. Judy Heidebrink. She is a clinical professor of neurology at the University of Michigan. She directs the Neurology Cognitive Disorders Clinic and is co-leader of the clinical core at the Michigan Alzheimer's Disease Research Center. She's been involved in collaborative clinical trials in dementia for over 20 years. In addition, she has led the University of Michigan's participation in the Alzheimer's Disease Neuroimaging Initiative. And with that, I will turn things over to Dr. Heidebrink.
Great. Thank you, Stephanie. Let me start by sharing my screen. And quick check, is everybody able to see and hear that okay?
Yes, we can.
All right, perfect. So as most of you know, there's been a lot of news in the past year coming out of clinical trials about new therapies for Alzheimer's disease. So I'm happy to share some of the highlights today. What I'm basically going to do is start with a little bit of background information and focus a fair amount of time on the anti-amyloid antibodies as therapies for Alzheimer's disease, since they have been getting probably the most news in the past year, but then also talk about some non-amyloid clinical trials. But to start with, let me just start with what is a clinical trial, because we talk about research coming out of a clinical trial. What do we mean by a clinical trial? Well, the definition of a clinical trial is a research study in which individuals are prospectively assigned, meaning they're systematically given a specific intervention with the goal of evaluating the health effects of that intervention. So it might be a medication, it might be an exercise regimen, but it's something that you're systematically offering or assigning someone to do so that you can determine the health benefits. And in many clinical trials, there's more than one intervention. There may be the active intervention, the study drug of interest, but there's also placebo. And that's really the best way of knowing the true effect of the intervention, like the study drug, because if you happen to see something while someone was taking a medication, it may be hard to know, is it truly the result of that medication, or was it going to happen just randomly by chance? But if you have a very similar group of individuals who are assigned to receive a placebo, then you can make more direct comparisons of the effect of the intervention compared to just what might have occurred simply by chance. Now, there are other ways to study potential therapies besides a clinical trial. So, for example, we hear news all the time about some type of lifestyle or medication that might be helpful as a therapy for Alzheimer's, but those aren't always coming out of clinical trials. If it's not a clinical trial, there may be more potential for bias. And what do I mean by that? Well, let's say, for example, you heard that people who drink hot chocolate are at lower risk of skin cancer. That sounds interesting, but the question is, well, how was that studied? Was it a clinical trial? Well, what if you found out that they surveyed people living all year round in Michigan and people living all year round in Florida and asked them how many cups of hot chocolate they had in the past year, and then also asked if anybody had been diagnosed with skin cancer? And you found out that people who reported drinking more hot chocolate were less likely to report having been diagnosed with skin cancer. Well, is it really the hot chocolate, or is it the fact that maybe more people in northern climates in Michigan were the ones drinking the hot chocolate and they don't have as much sun exposure compared to those in Florida? So to really know, was it the hot chocolate, you'd want to do a clinical trial where you take individuals, regardless of whether they live in Michigan or Florida, and assign them to either drink hot chocolate or maybe drink a different hot beverage, hot tea, or maybe a placebo beverage, and then follow them over time to see if there's a true difference in terms of the development of skin cancer. So it's always helpful when you hear something about a potential therapy to know, Is it coming from a clinical trial or some other type of clinical research? So today I'm going to focus on clinical trials. And just by way of background, if we're thinking about a medication that might be a potential therapy for Alzheimer's disease, for example, typically that goes through a series of phases of clinical trials that focus on different elements of that particular treatment. So the early trials are called phase 1 trials. They might involve very few individuals, and they're really looking at whether the medication is safe, how much people can take, how long does it last in their system, so how frequently will they need to take it, is it a daily medicine, a weekly medicine? So a lot of initial information about the drug itself, and particularly focusing on its safety. If things look fine with the phase 1 trial, then phase 2 trials are done, again, gathering more information about safety, because often these are given for a longer period of time, but starting to look at the potential benefit. Is there a promising effect that would look like it could be useful to treat a given disease? And then if that looks promising, then studies will move to the phase 3, and that's really kind of the make or break. What is the overall risk and benefit of this particular intervention? Does it look like it is helpful in treating the condition of interest? I'm going to focus a little bit on the phase 3 trials, because those are the trials where the drugs are closest to being available in terms of getting approval by the FDA, for example. But there are lots of trials in earlier stages of compounds in earlier stages of development too, and I won't be able to talk about all of them today, so I'm going to focus again on sort of the newer available therapies and their clinical trial data. And just as a reminder, I'm focusing on Alzheimer's disease, but that's not the only cause of dementia. Dementia is really just a descriptive term that indicates someone has had a decrease in their thinking abilities that's interfering with their day-to-day function. But Alzheimer's isn't the only possible cause of dementia. It happens to be the most common, but there are other not infrequent causes that are shown here in this slide. And often people may have more than one cause too. In clinical trials, they'll often try to identify individuals who only have the one disease of interest, but that can be tricky as well. But often the drug might be studied because it has properties that may be helpful in one specific type of dementia because of the brain changes that are going on. Or sometimes drugs are studied that may have more broad implications to help different types of dementia as well. So I'll try to be clear about when I'm talking about an Alzheimer's disease trial, or whether I'm talking to one about more broadly that might help with memory in general or dementia in general. So let me start with the anti-amyloid antibodies. This is just a slide showing some of the different anti-amyloid antibodies that have been studied in phase 3 clinical trials. Why might they be a potential option for treatment for Alzheimer's? Well, as you may know, in Alzheimer's disease, there's an abnormal accumulation of the amyloid protein. It starts as sort of smaller fragments, but those fragments start to build up into larger and larger aggregations that eventually become the amyloid plaques that are one of the pathologic hallmarks of Alzheimer's disease. And so researchers have designed compounds, antibodies, that can bind to the abnormal forms of amyloid to help the brain remove that amyloid as it's accumulating in the brain in someone with Alzheimer's disease symptoms. So these are called monoclonal antibodies, or MAB. You'll see at the end of each of their names. So if you see a drug with MAB, it means it's an antibody directed at something. Doesn't necessarily mean it's an anti-amyloid antibody, because there are monoclonal antibodies to treat other diseases, but here are just some of the ones specifically targeting amyloid. So the first question is, well, do we know that they can do that? Do we know that they can get into the brain, bind to the amyloid, and help remove the amyloid buildup from the brain in individuals with Alzheimer's disease symptoms? And the answer to that is yes, at least for some of these compounds. So for example, a drug called aducanumab was studied in individuals with relatively mild symptoms of Alzheimer's disease, either people who had a mild degree of dementia or something called mild cognitive impairment, where they were still independent in their day-to-day functioning, but were experiencing memory changes that were beyond those of just normal aging. And all of these individuals had evidence of abnormal amyloid accumulation in the brain. So one of the signatures of Alzheimer's. And that was detected by specialized brain scans called PET scans, using a tracer that specifically binds to the amyloid protein if it has built up in the brain. So the left column are just PET scans from 4 individuals at the start of this study. The tracer, when it binds to amyloid, if there's amyloid there, shows up as the brighter kind of reddish orange signal. So you can see all of the brains had evidence of amyloid buildup. And in this initial study, individuals received monthly infusions of either a placebo or the aducanumab antibody at different doses. And they had PET scans at the beginning of the study and at the end of the 1 year of the monthly infusions. And you can see for this individual who was on the placebo, they still have evidence of lots of amyloid buildup in the brain, really no change, or if anything, slightly darker red in a few areas. Whereas the individuals who receive the active drug, you can see less evidence of amyloid buildup. And in fact, the individual who received the highest dose on a milligram per kilogram basis had a PET scan at the end of the study that you could argue was normal. There wasn't any of that red, bright area, any significant area of amyloid binding. So we know this drug can get into the brain and help remove the brain amyloid. Another drug, lecanemab, very similar study was done looking at different doses of the drug in individuals with early symptoms of Alzheimer's disease and evidence of that brain amyloid accumulation. They didn't publish nice pictures, unfortunately. They did a graph instead, but it shows really the same effect, that if you receive the placebo, there was really no change in the amyloid level from the beginning to the end of the 18-month study. But if you got the active drug, you saw a drop in the amyloid level in the brain, particularly if you received a higher dose at a more frequent interval. So people receive different doses and receive them either twice a month or once a month. But the group that had the greatest drop in that abnormal brain amyloid was the group who received the highest dose most frequently, that 10 milligram per kilogram every 2 weeks, essentially, or twice a month. So both of these drugs, aducanumab and lecanemab, have been granted what's called accelerated approval by the FDA because of this biological effect, because of their ability to remove brain amyloid. But the bigger question has been, are they helpful clinically? And only one of the two drugs, lecanemab, has received full FDA approval because it not only has biological effects, but there's been convincing evidence of a clinical benefit when given to individuals with early symptoms of Alzheimer's. So this drug just received full FDA approval a few months ago in early July, and it was largely based on a large phase 3 study where roughly 1,800 individuals with symptoms of Alzheimer's disease and evidence of abnormal brain amyloid received that highest dose, the 10 milligram per kilogram infusions every 2 weeks of either the active drug or a placebo. And the main outcome that they looked at clinically was something called a clinical dementia rating scale. This is a numerical rating of someone's level of impairment that is derived from an interview with the individual with Alzheimer's symptoms and with someone they know well, their study partner. And they're assessed for their ability to remember things, their awareness of things like time and place, their reasoning, their ability to do things at home and out and about in the community, and they're rated in terms of their level of impairment. Now, if you track how their level of impairment changed over time, if they move down on this graph, that means a higher degree of impairment or less independence. And you can see that, unfortunately, both groups did decline, whether they received the active drug or the placebo, but that decline was clearly less in the individuals who received the lecanemab compared to the placebo. Numerically, that decrease in function and cognition was 27% slower if they received the lecanemab. It's hard to know what exactly that means, but one way of thinking about it is at the end of the trial, the individuals who had received the lecanemab were essentially functioning and thinking about where the individuals on the placebo group were about 6 months earlier. So in a sense, buying you 6 months more time to be more independent over that 18 months. So that's another way of sort of thinking about the clinical effect. And it wasn't just this one measure. They looked at other measures to determine the effect of lecanemab. The biggest effect was clearly the effect on the brain amyloid. So in this study, again, if you receive the placebo, levels of brain amyloid stayed relatively stable, but a sizable drop, so that a significant portion of individuals at the end of the trial had essentially a normal PET scan. But if you look at clinical measures, you also see a difference between the lecanemab group and the placebo group, whether it's a measurement of thinking or whether it's a measurement of their day-to-day function, how independent they are. And again, we're seeing a difference where those receiving the active treatment are functioning at a level where the placebo group was somewhere between 3 to 8 months earlier, depending on which exact scale that you use. So evidence of slowing the clinical progression, not just removing the brain amyloid. Well, is there a downside to receiving lecanemab? Is there a potential for side effects? Well, as we know, all medications have some risk of side effects. What side effects were observed in the large phase 3 trial? Well, two main types of side effects. One was an infusion reaction. You're introducing this antibody intravenously. It's a foreign substance, and people can have a reaction to that, just like you might get a temporary reaction after you receive a vaccination. You feel kind of achy the next day or soon after receiving it. So at the time that people receive the infusion, about 1 in 4 did experience some flu-like symptoms, kind of achy chills, things like that. And mostly this was with the very first infusion. It wasn't something that typically continued on with multiple infusions. And it was typically quite manageable, either slowing down the rate of the infusion or treating with over-the-counter medications, things like acetaminophen, Tylenol, or diphenhydramine. So pretty much a temporary reaction to the initial infusion. The other typical side effect that we know can be associated with these antibodies are what are called amyloid-related imaging abnormalities, or ARIA is the abbreviation for that. And they're called imaging abnormalities because they're typically picked up on something like an MRI scan. You can identify one of two types of ARIA. There's ARIA-E, which stands for edema, which can be evidence that fluid is getting into the brain. So a little bit of swelling or edema can occur. Or the other type of ARIA is ARIA-H, which stands for hemorrhage. There can be little bits of leaking blood coming out of the brain when people receive some of these antibodies, because as the antibodies are getting into the brain, binding the amyloid, pulling it out, the blood vessels can become a little leaky. So fluid can go in and lead to evidence of swelling, or tiny amounts of blood can leak out. Now, fortunately, either of those, ARIA-E or ARIA-H, is typically silent, meaning an MRI scan might pick it up if it's there, but the individual may not experience any symptoms whatsoever, particularly if the areas that show the ARIA-E or ARIA-H are relatively small. Often the ARIA-H is almost like the tip of a pen. That's how small those little areas of bleeding can be. But in some cases, it can be more significant, and depending on where the ARIA occurs, people can experience symptoms. If there's just general swelling, it might be headache or nausea. If the swelling is more related to parts of the brain that are important for vision, there might be changes in vision, or in some cases, seizures can occur too. So all individuals receiving this medication need to have an MRI at the beginning to know if they have anything that would make them at higher risk for the ARIA in the future. And also they need to have regular MRI scans at set intervals to look for evidence of ARIA in case the medication needs to be held temporarily or permanently, depending on whether that ARIA is observed and to what degree. And most cases of ARIA tend to occur earlier with the infusion. So the first 6 months in particular are where the monitoring for ARIA is more important. It turns out that not everybody is at equal risk of developing ARIA. There is a genetic risk factor. It's a risk factor for Alzheimer's disease, period, but it's also a risk factor for the development of ARIA with lecanemab and some of the similar medications. So the apolipoprotein E gene, or ApoE gene, is a gene we all carry. It comes in different types, type 2, type 3, and type 4. We have 2 copies, one from our mother, one from our father. They can be the same, they could be different, just depending on what types our parents have and what we've inherited from them. Having one or more copies of the E4 gene is a risk factor for developing Alzheimer's disease over your lifetime. But in individuals who have Alzheimer's disease, it's also a risk factor for developing the ARIA when given lecanemab. Probably because if you have the E4 gene, there's a little more of the amyloid around blood vessels compared to people who don't have the E4. So those blood vessels become a little more vulnerable. So the overall rate of ARIA-E, the swelling in the lecanemab phase 3 study, was about 13%. Again, very frequently occurred without any symptoms. Only 3% of people had symptoms. But if you look based on someone's genetic makeup, if they didn't have any copies of the E4, a lower overall risk, but if you had one or more copies, particularly the people with two, a higher overall risk and a higher risk of symptoms as well. Same was true for the ARIA-H. It rarely caused symptoms to begin with, so they didn't break it out further, but the risk of just seeing that ARIA-H on a scan was definitely related to how many copies of the E4 gene someone carries. So it's important that if anyone is considering this therapy, to have a genetic test to know whether they may be at higher or lower risk of this particular complication, because this drug is now available to treat individuals with early symptoms of Alzheimer's disease and that confirmatory evidence of brain amyloid. But again, it's given as an infusion every 2 weeks, which is logistically challenging. So what might be on the horizon in the future? Well, very recently it was announced the results of some early studies using an injectable form of lecanemab, not as an intravenous infusion, but as shots that are given weekly under the skin, like someone might receive an insulin shot, for example. And so individuals who were participating in the original study but didn't get the active drug, they got the placebo, some of them were offered the chance to get the active drug in a subcutaneous injection form, and they looked at their PET scans 6 months later and compared to the individuals who'd received the IV form in the original study, there was an even greater degree of amyloid plaque removal. There was also a lower frequency of injection site reactions. You're not giving an infusion now, it's just a shot under the skin. But the rates of ARIA look to be similar or perhaps numerically slightly higher compared to the rates with the intravenous form. Now, again, it's hard to make direct comparisons of these numbers because this is a relatively small number of individuals receiving the subcutaneous. So if we got up to 1,800 individuals like the overall Clarity trial, we might have a better sense of whether those ARIA rates are truly going to be different. But obviously, this is a much more accessible way of receiving this drug if it could be self-administered at home through an injection. So they do plan to submit more data to the FDA so that this might potentially be a treatment option in the future. Now, the other drug that's gotten a lot of news recently is sort of the new kid on the block, the newer monoclonal antibody called Donanemab. It is not yet FDA approved, but we anticipate that the FDA will review it probably early next year because it similarly had a large phase 3 trial with about a similar number of individuals with early symptoms of Alzheimer's. It wasn't exactly identical to the lecanemab trial. They did a couple interesting twists. Not only did everyone have evidence of amyloid buildup, such as through an amyloid PET scan, but they also did tau scans to look at the level of tau buildup in the brain. And they wanted to focus in particular on individuals who had relatively lower amounts of tau, with the thought being they might still benefit from an amyloid-based therapy if there isn't as much tau spread already at the time that they begin treatment. But they did also include individuals with a higher level of tau to see if there would still be an overall benefit, kind of regardless of the tau scan result. And with this study, they received infusions of donanemab or placebo every 4 weeks, so a little bit different administration schedule. And they also looked at PET scans every 6 months, and they stopped giving the individuals who were on the active drug treatment arm, they stopped giving the donanemab if the PET scan had showed clearing of the amyloid. So some people received the active drug all the way through the 18 months, but some people, in fact, about 2/3 of individuals, came off the drug early. They were continuing to be followed. And again, they didn't know whether they were receiving the active drug or placebo, but they didn't actually need to continue on the active drug because there was amyloid clearing, but they were still followed to see what the effect was up to 18 months. And if you look at the population specifically with the lower levels of tau in the brain, they chose a slightly different primary measure of clinical effect, but they also looked at the clinical dementia rating scale too. So I put both of these outcome measures on the graph. Compared to the placebo group, those who received the active donanemab had about a 36% slowing. Again, if you look at the graphs, you're seeing that the treatment group is behaving about where the placebo group was roughly 6 months or so earlier. If you look at overall, regardless of their tau scan, it's a similar result, but the benefit's not quite as robust. So you can see the curves, just looking at people with lower levels of tau versus higher levels of tau, there's a bit more decline when people have higher levels of tau overall, but still that decline is slower if they receive the active drug. And if we looked specifically at the individuals who ended up coming off the infusions, maybe at the 12-month point, because their PET scan showed clearance of the amyloid, they still showed that ongoing benefit. There was an ongoing separation between the donanemab group and the placebo group. And these curves, if you look at them, they look like they're getting a little wider as time goes on. So that benefit might be even greater past the 18-month point. But we don't know that because the study ended at that point. But it's possible that this separation is just the start of the benefit, not the maximum benefit of this therapy. There were some infusion reactions, a little bit less frequent in this study, but there was also ARIA that was a little bit more frequent. So the evidence of ARIA-E was about twice as often, 24%, about twice as often. It was symptomatic as well compared to lecanemab. But again, a different group of patients based on how they were recruited in the study because they needed to have evidence of tau changes too. And there was ARIA-H reported as well, including during the trial, 3 deaths that were felt to be related to receiving the drug. And in some cases, it was an individual who had evidence of leaky blood vessels even before the study started. So it's possible if the FDA reviews this drug and approves it, they might say any abnormal changes like that on an MRI scan would disqualify someone from potentially receiving this therapy. So there may be some ways to make that safety even greater by really looking at who was at risk for the more serious forms of ARIA. So if we just sort of summarize, what do we know? We have 2 drugs that have been approved based on their biological effect, that's Aducab and lecanemab. Lecanemab approved based on its clinical effect, and donanemab likely to be approved in the coming months. What do they show, particularly the lecanemab and donanemab? Well, they do show a benefit in individuals with early symptoms of Alzheimer's disease. Roughly, they may slow that progression by somewhere around 30%, kind of buying people 6 or so extra months of greater independence. That benefit may be larger as time goes on. We don't know, but the graphs are encouraging. And these are really the first therapies that directly target one of the hallmark pathologies. They really modify the disease. They remove that amyloid from the brain. So those are the positives. The risks, though, you can develop those ARIA-related imaging abnormalities. And in particular, individuals who have the E4 gene, if they've got 2 copies, that risk is much greater. And at least in some of the studies, people who are on very powerful prescription blood thinners were at higher risk of developing side effects. So for lecanemab, for example, it's not recommended that people who are on very powerful prescription blood thinners be candidates for lecanemab therapy. And donanemab, that will need to be a consideration as well. You do have to have evidence of brain amyloid, which in the studies was typically a PET scan paid for by the sponsors of the study. But in clinic, we really haven't had the opportunity to get routine brain amyloid scans because they haven't been covered by insurers. That, however, looks like it is changing. Just last month, Medicare removed essentially a restriction on their coverage for brain amyloid imaging. Previously, they said they would really only cover it in the context of a clinical trial, but now they've removed that restriction. So we're hopeful that we'll be able to use brain amyloid scans now in the clinic to really know if someone has Alzheimer's changes in the brain and would they be an appropriate candidate for this type of therapy. At present, these antibodies are received through regular intravenous infusions. Again, there may be hope of the under the skin injections in the future. They're new drugs, they're expensive, and if they do need to have regular infusions, it's not just the drug expense, it's the logistics and expense of getting the infusion either on a every 4-week basis or every 2-week basis, depending on which drug we're talking about. And we don't know precisely how long to treat someone. Can you just stop when the amyloid PET scan looks normal, such as with the Donanemab trial, or will that symptomatic benefit fall over time and they'll need to be redosing in the future? So we don't know all of those details yet, but very exciting, and really the latest therapy. It's the newest therapy that we've had now in almost 2 decades as a treatment for Alzheimer's disease. Now, I mentioned these have all been studied in people with early symptoms of Alzheimer's, either people in the mild dementia stage or in the mild cognitive impairment stage of Alzheimer's. But it turns out if we can look into the brain even earlier, those changes, particularly the buildup of that brain amyloid protein, is actually starting many years before even the earliest symptoms begin. And so even in the studies of lecanemab and donanemab, we're already lagging our treatment several years behind that amyloid buildup. They've done some sub-analyses suggesting that if you look at the data, folks who had the mildest symptoms, and perhaps also younger individuals, again, who may be at earlier biological stages, seem to have the greatest benefit. So what about turning the clock back even further? What about treating people who are in the phase of silent brain amyloid accumulation? Would these therapies even be more beneficial in halting the risk of Alzheimer's disease symptoms as opposed to simply slowing it down once those symptoms are already there? Could they be used as preventions? Well, that was exactly the idea behind a large Alzheimer's prevention trial using a monoclonal antibody called solanezumab. The study itself was called the A4 study, and we were one of the multi-center participating sites in the trial. But unfortunately, with data that was just announced this year, there wasn't a clinical benefit. So people didn't behave differently whether they received the solanezumab or a placebo in terms of reducing the risk of cognitive decline in individuals who were cognitively normal at the beginning of the study, but had evidence of the brain amyloid buildup. Well, it turns out, unlike the antibodies I've talked about so far, solanezumab did not lower brain amyloid as measured by a PET scan. So here are just graphs from the average PET scan from individuals. If you receive the placebo group, the amyloid levels just crept up gradually over the several years of the study, maybe a little less buildup if the solanezumab, but not the big drop that we were seeing in the trials I showed you with donanemab and lecanemab and aducanumab. And so it might have been the right idea, but just not the right drug, because it turns out if you looked at the amount of amyloid someone had in their brain at the beginning of the study and divided that into thirds, people at the highest level, the middle third, and then the lower level of amyloid buildup, that did seem to predict whether they would experience a change in their thinking overall. So those with the highest level of amyloid to begin with, they tended to show a greater downward trend in their thinking scores compared to individuals with lesser degrees of amyloid buildup. So still suggests that amyloid is important, and if we can really lower it early on, it may be useful as a prevention therapy. So the drugs now that are being used or might soon be used clinically in individuals who already have symptoms are being studied in prevention trials. So there's a prevention trial using donanemab, and we at the University of Michigan are participating in a prevention trial using lecanemab. So it's looking for individuals who might be at risk of having that silent buildup of amyloid in the brain just based on their age or maybe their family history. So we're looking for individuals between ages 55 and 80 who are thinking normally, but show evidence of that silent amyloid buildup by a PET scan. And depending on how much buildup is there, they will be assigned to receive either lecanemab or placebo, either every 4 weeks if they have lower levels of amyloid buildup, or every 2 weeks, at least initially, if they have higher levels. Then after a couple of years, we'll drop back to every 4 weeks. Again, trying to drop that amyloid early on based on how much is there and seeing if that protects individuals from developing symptoms later on. And so everyone will have a PET scan, but they're actually going to have a blood test first because we can measure minute levels of the amyloid and tau proteins in the blood not with perfect accuracy, but reasonable enough accuracy to use that as a first step to decide who should go on to have a PET scan. If the blood test says the likelihood of us having a change on a PET scan is so low, someone might not move on to the PET scan stage of the study. But ultimately, they do need to qualify based on having evidence of amyloid buildup on a PET scan, and they'll be tracked with measurements of their thinking and daily function as they receive the lecanemab or the placebo to see if this is useful as a prevention therapy. All right, so that's the kind of update on the amyloid, the anti-amyloid antibodies in a nutshell. Let me switch gears though and talk about other potential treatment avenues. One would be treating the other main pathology of Alzheimer's disease, the tau that builds up in the form of tangles. It turns out, as I've mentioned, you can have a lot of amyloid building up in the brain silently without symptoms or before symptoms develop. But once people develop symptoms, the amount of tau and where it is found in the brain tends to correlate better with the progression of those symptoms. And so it tends to become widespread a little bit later compared to the amyloid. So you might be able to intervene more successfully once people have already developed symptoms, whereas amyloid might be more successful very early or even on a prevention basis. There are antibodies directed at the tau protein that are going through clinical trials. Some of the initial ones did not show early promise, but there are some newer ones that are making their way through those early phase trials. But there are also some very interesting non-antibody approaches to tau therapies. One is a genetic approach to essentially silence the gene that produces the tau protein. And so there's something called an antisense oligonucleotide, which basically just binds to genetic material like RNA and prevents it from being translated into a protein. It's like if you had a recipe for something and you took a sticky note and covered the recipe, so the recipe couldn't be used. Now, because you've got to treat the tau protein in the brain, this compound has to get into the brain, and it's not something you can just take by mouth. It in fact has to be delivered sort of into the spinal fluid. So people had to undergo lumbar punctures to deliver this compound that gets into the brain and potentially blocks the genetic translation of the tau protein. So the hope is if you measure tau buildup in the brain, you might be able to prevent further buildup by blocking that gene translation. And so in this very preliminary study, individuals received 6 months of placebo, and they had PET scans before and after. So the first two scans here on the left are at the start. And then after 6 months of placebo, the tau binding is the brighter area. So it looks about the same after 6 months, maybe even a little bit more. And then they got 1 year of the active treatment. The hope was that you wouldn't see increased evidence of tau. And what you actually saw was a decrease. So not only did it seem to prevent further buildup of tau, but actually seemed to be successful in reducing tau buildup in the brain. Again, a very preliminary study, small numbers of individuals. They've just started to look at whether there might be a clinical benefit. So too soon to really weigh in, but this may be a potential therapy. Again, logistically challenging to deliver, but it may be something where they can find better ways of getting it into the brain. And so some people have proposed, why not do some combination therapies? Let's target amyloid and tau at the same time. And so a group that we're involved with called the Alzheimer's Clinical Trials Consortium has proposed what they call a tau platform design, where they may study simultaneously more than one therapy directed at tau, but also either in comparison or in conjunction with therapies aimed at treating amyloid. So maybe the two together are better than either one alone. I know this slide is a little fuzzy and maybe hard to read. In fact, that's just because that's the best slide I could get. But in fact, the details of this platform trial are still a little bit fuzzy because it was proposed before these monoclonal antibodies became clinically available. And so the question is, would it be ethical to use a placebo group in this study if people are eligible to receive lecanemab or in the future donanemab, or should everybody in the tau studies also get an amyloid treatment as well? So the details are still being worked out, but it's encouraging that we may be able to approach this disease by treating more than one aspect simultaneously. And that's just the amyloid and the tau part of Alzheimer's disease. There's a lot more that goes on in the brain besides just the buildup of the amyloid plaques and the tau tangles. So this slide just says, shows all the different things that may be happening behind the scenes. It's not just that there's amyloid building up in plaques and tau and tangles, But there's inflammation that happens when these changes are occurring. So anti-inflammatory treatments might be successful. And these proteins, as they build up, bind to a variety of receptors on the nerve cells that might mediate how they injure the nerve cells. And so potentially interrupting that binding might be a way. So not directly targeting the amyloid or the tau, but interfering with how those proteins interact with the nerve cells and do their damage. And so one compound goes by the name CT1812, looks like it can do that. It can prevent some of these toxic proteins from binding at something called the sigma receptor. And by doing so, it may be able to prevent the injury to the nerve cells that occurs when these proteins are binding to the sigma receptor. So these are pictures just from a basic science experiment. The nerve cells are shown in green in the lab. And so they have these kind of long arm-like structures, and the toxic protein is shown in red. And so on the left-hand side, in a kind of experimental model of a brain disease like Alzheimer's, you can see a lot of binding of the toxic protein to the nerve cells. But if you then introduce this compound, you see the nerve cells really don't show any of those red blobs on the nerve cells anymore. So you're eliminating or blocking the binding. And so we hope to very soon launch a trial that is looking at this compound to see if it is a potential therapy to treat Alzheimer's disease. So it's going to be focusing on individuals with early symptoms of Alzheimer's who do have some biological evidence of changes in the brain, either an amyloid PET scan or spinal fluid changes. And they'll be randomized to receive either one of 2 doses of the experimental drug or a placebo, and tracked roughly every month. People will come in for visits over a period of 1.5 years to see if the compound is safe and to see if it is helpful as a treatment for Alzheimer's. Now, I lied a little bit when I showed you this slide because the protein that actually is shown in the slide is not amyloid protein. It's actually alpha-synuclein protein, which builds up in Lewy body dementia. So this compound may also be a potential therapy for Lewy body dementia because it may have a similar effect in preventing binding of its toxic protein in Lewy body dementia. So there's another smaller scale trial with the same drug that's been launched to look at as a potential therapy for Lewy body dementia. And there are other even more sort of gentle therapies that are being explored, such as vitamin supplementation. So there's a particular vitamin called thiamine, or B1, that's important for a lot of brain processes. And we know that individuals who have severe thiamine deficiencies can have associated memory loss. It turns out that thiamine isn't necessarily easy to get into the brain, but there is a compound called benfotiamine that becomes converted in the brain to thiamine. So it looks like it's easier to take it in the pill form and then get converted into the brain into thiamine. In a very small study, this compound suggested a benefit. So one of the other studies that we are just about to launch is a trial of benfotiamine in individuals with early Alzheimer's disease symptoms. And for this, we're looking at individuals who have one of the blood tests consistent with Alzheimer's changes in the brain. And individuals will receive either the benfotiamine or placebo for about 18 months and be followed to see if this drug is safe and hopefully a helpful treatment for Alzheimer's disease. Well, you may have said, well, gosh, I think I heard something on the news about the benefits of vitamins. What was that? Well, there have been a couple announcements about multivitamin studies, and these aren't specific to Alzheimer's disease, but they're just looking to see, do they help someone as they age, potentially protect them from thinking changes or other health changes. So a large trial called the Cosmos trial, because they were studying cocoa supplements, so flavonols in cocoa extract, along with multivitamins. So the acronym cocoa supplement and multivitamins outcome study is the Cosmos trial. Very large study looking at older individuals who received either the cocoa extract or a multivitamin or both or placebo over several years. And a subset of individuals were tracked with tests of their thinking, either through telephone interviews or through web-based testing of their memory and thinking. And in those sub-studies where they did cognitive measurements, the individuals who were receiving the multivitamin seemed to show better cognitive scores over time compared to the individuals receiving the placebo. And unfortunately, the cocoa extract didn't have a benefit, didn't have an overall benefit to in terms of heart health and other things that they're looking at overall. but the amount of benefit has been a little bit questionable. For example, this web study, the difference that they showed between the multivitamin and the placebo group was on a specific task where they had to listen to a list of 20 words, and right away after, actually they were shown on the screen, right away after they saw this list of 20 words, they were asked to type in as many words as they had just seen. And then later on, they were asked to type in those words later to see if they could retain those words. So it's a memory test. But the difference between the multivitamin placebo group was only when they initially typed in the words just after seeing them. It wasn't on the later ability to remember them. And the difference was pretty small. So at baseline, if you were receiving the placebo or multivitamin, you were able to type in about 7 of the 20 words right away. When they tested a year later, technically the multivitamin group did type in a few more, like almost 8, but about 7.5 in the placebo group. And if they tested 2 years later and 3 years later, they weren't really distinguishable. So it was a pretty small effect, and it's hard to know whether that's really clinically meaningful. But I guess the good news was there weren't any untoward side effects of taking a multivitamin supplement. So, something to consider. Other types of therapies, non-medication approaches that have gotten attention have been things like treating hearing loss. There is an expert panel that meets roughly every few years to review potential risk factors for dementia that might be modifiable, things like other health conditions or alcohol consumption, smoking, head injuries, things like that. And among the potential modifiable risk factors, one that was identified as having the greatest potential impact, if addressed, was hearing loss. So individuals with hearing loss do seem to be at higher risk of dementia. But does that mean if you treat the hearing loss, you can change their trajectory? Well, potentially. A recent study took individuals with hearing loss. They recruited them from 2 different pools of volunteers. One were relatively healthy subjects. Others had a higher risk of cognitive decline because they had other risk factors. And both of these pools of individuals were randomly assigned to either get hearing aids or just general health education. And if you look specifically at the high risk group, if they received the hearing aids, they had less cognitive decline over the 3 years that they were followed. Healthy volunteers didn't necessarily seem to make a difference, but again, they might not have been as much at risk for cognitive changes to begin with. So maybe in particular targeting therapies to those at greatest risk. What other types of non-medication therapies might be helpful? Well, very recently announced study looked at Tai Chi. There have been previous studies looking at the potential health benefits of exercises, including ones like Tai Chi, where there's an emphasis on the mind-body connection. Well, they put a little different twist in this study that individuals here were all recruited because they had memory concerns, and their average score on a memory test was just a little bit below what might be considered the cutoff for normal thinking. So they were just a little bit lower, 25 out of 30. And they were assigned to either do stretching exercises, kind of a standard Tai Chi regimen, or something they called cognitively enhanced Tai Chi. This was where, in addition to doing the regular Tai Chi moves and poses, they might do something else at the same time. Maybe they had to associate a number with a specific Tai Chi form, or they had to spell things while they were doing their Tai Chi exercises. So there was a more enriched cognitive component. And these interventions were all delivered virtually through a video format, because this study took place in the time of COVID. And it was only an hour twice a week for 24 weeks. And they looked at how they're scored on this cognitive test. And if they were engaged in the Tai Chi compared to those who just did the stretching, their MoCA scores bumped up on average 1.7 points. But if they got the enhanced Tai Chi, it was 3 points higher. And they also did better if they asked them to do tasks like walk and do subtractions at the same time, kind of a multitasking. People didn't slow down as much when they were doing the mental subtraction task while walking if they had participated in the cognitively enhanced Tai Chi. So this might be something that potentially reduces the risk of falls too. So there may be plenty of benefits to this mind-body connection, particularly when there's an additional cognitive enhancement. So these last 2 studies just emphasize that doing sort of multimodal approaches, not just a single thing, not just a physical action, but a mental action, treating hearing loss, treating multiple things together, particularly targeting those individuals who might be at greater risk, might give us the best bang for our buck in terms of preventing dementia. And that's the approach taken by another study that's ongoing. It's only being done at select sites in the United States, so we're not involved in this study, but it mimics a similar study that was done in Scandinavia, the Finnish study targeting older adults. It was called the Finger study because it was Finnish and looking at sort of a geriatric population. But they're targeting individuals who might be at risk because of their age or a relatively sedentary lifestyle or certain risk factors. And they're either giving them kind of general guidance about healthy lifestyle and behavior versus a more structured intervention where they get specific guidance on exercise, nutrition, management of other health conditions, and they'll be tracked over time to see if these can, in a positive way, impact their cognitive function, as well as a lot of other potential health outcomes. So just again, the idea that approaching things in multiple ways at the same time may give us the most bang for the buck in terms of dementia prevention. So let me just again circle back to what we're doing here at the University of Michigan. And these are only the studies that I've been involved with. There are plenty of more through our Alzheimer's Research Center. So I encourage anyone who might be interested to look at our Alzheimer's Research Center website for research opportunities. Some of the ones, we still have folks who were in an earlier phase of lecanemab trial who are still being studied. But the ones that are currently recruiting again is the lecanemab prevention trial, that AHEAD study. There are other studies where people are just simply followed over time to track their thinking abilities. And then I mentioned some that we're just about to launch, the CT1812 trial, the benfotiamine, and hopefully probably sometime next year, that tau platform trial. So with that, I will stop and I'm happy to answer questions.
Yeah, it looks like we do have a question in the Q&A. It says, do you prescribe the anti-amyloid antibodies for your own non-research participants?
So that, yes, the answer is yes. Now that lecanemab has received full approval by the FDA as of July, that also means that Medicare, who had previously said we will only pay for these, we will only cover these if they receive traditional approval, that means Medicare has agreed to pay for them. So we are currently evaluating anybody who's coming to our clinic to determine if they're an appropriate candidate for lecanemab, and the same will apply if donanemab gets approved as well.
And can you speak to the controversy from the FDA advisory CTE, who was against FDA approval?
So I don't know if that's referring to the previous drug, aducanumab. I would say that's the drug that had a lot of controversy because it received what was called accelerated approval. So ultimately was approved based on its biological effect, not based on its clinical effect. But there was a big controversy about should it have been approved at all? And in a sense, it wasn't submitted for accelerated approval. It was submitted for general approval, and they kind of shifted the target. And there was an external advisory committee, a panel of experts who weighed in, who sort of uniformly turned down. They were asked to look at it from a traditional approval standpoint, and they uniformly said, we don't think the evidence is there yet. In contrast, lecanemab had an external advisory approval review board that really universally said, we think this drug ought to be approved. So big difference in terms of the FDA approval process between aducanumab, which only got accelerated approval, and lecanemab, which now has full approval.
Okay, I had another person say, I have a possible person which I think would fit CT 1812. He is going to turn 55 and it is his short-term memory.
Mm-hmm. So yeah, anybody who thinks they might be interested in any of these clinical trials, like I said, some of them we're ready to enroll folks right now. Some were just in the final preparation stages. When we are ready to enroll, they will typically be posted on the Alzheimer's Disease Research Center website. So you can click on the tag that says research, and it will open up a list of enrolling studies. So you can see the AHEAD study, that prevention trial is listed there, many other studies as well. So again, I've only focused on a few. The CT1812 trial, the START trial, we'll get there as soon as we're ready to start enrolling. And when we list the studies there, we list the primary contact person. So if you say, hey, I'm interested, who do I need to speak with? It's listed there too.
Yep. And I am going to put our website in the chat. So,
Great.
all right. Are there any treatments on the horizon for patients beyond the early stages of Alzheimer's?
That's a really good question. And many of the trials are focusing on early symptoms because the concern is, particularly if it's an amyloid-based therapy, the benefit just may not be there with people in later stages. But the tau therapies, I think, have the potential at least. And there have been others that are more neuroprotective or neurorestorative that could potentially be used at a later stage. So some trials are starting to target later stages. Some of them are symptomatic trials too, looking at behavioral complications, which can occur across the spectrum of severity. There's an interesting trial looking at the ingredients in marijuana, THC, CBD, as being potentially helpful for agitation near the end of life in individuals with dementia too. So the goal is really to offer therapy sort of across the spectrum, but again, it will depend on what the drug is targeting and whether it makes sense to enroll individuals at early versus later stages in terms of where you think the benefit's most likely to occur.
All right. Do you know about any interventions targeting glymphatic transport and or vascular health for improving clearance of proteins?
Yeah, so that's kind of a big, broad question. And it's an interesting thing that, these proteins, particularly the amyloid, it's a protein we all have in our brain, but it doesn't usually accumulate in the form of these plaques, at least in most individuals. And so how does our brain normally process amyloid? And there's this idea that it kind of maintains in circulation or turns over through something called the glymphatic system, kind of like the lymph system, sort of a drainage system, and that system may be more active at certain times of day, like at night when we're in deep restorative sleep, which has interestingly led to links between poor sleep or ineffective sleep and a risk for Alzheimer's disease. So in a sense, targeting sleep may be a way of treating that lymphatic system. And there are other approaches that are being looked at, and certainly vascular too. I mean, even things like simple blood pressure medicines are being looked at as a potential therapy, not just because of protecting the brain from high blood pressure, but because they may have other mechanisms that are more directly relevant to Alzheimer's. So yes, other therapies like that are being looked at.
How do we get an appointment for you to review a person?
So if anybody's interested in having their care through our cognitive disorders clinic, we don't have a we're not specifically evaluating people only because of lecanemab. We're evaluating anybody who wants to have care. Basically, it takes a referral from a physician to our clinic. And if you have any questions about that process, you can always call our clinic. I can say the number, we can put it in the chat, but it's area code 734-764-6831.
All right. Do you have any people in the STEM study? What kind of results?
Yeah, so the STEM study is really run by my colleague, our clinical core leader, Ben Hamstead, neuropsychologist. That's probably a whole separate talk, which would be excellent to consider as part of this series because there are a variety of STEM studies. And in fact, he's got offshoots, ones for Alzheimer's disease, ones for the more typical presentation with memory loss, ones for the atypical presentations, people who have predominantly language symptoms, because the idea is it's a study of electrical stimulation of the brain, and where you target it might need to be individualized. So targeting the brain regions involved in memory in someone who has predominant memory loss, or the language centers in someone with predominant language. So a variety of these stimulation studies are underway, and some are also being targeting the non-Alzheimer's dementias too. So I think that would be a great opportunity to have him come and talk about his research. You can find out more about enrolling in one of these studies on the Alzheimer's website.
All right. What did the earlier Alzheimer's treatments do, and how different were the results compared to lecanemab?
So I don't know if that's referring specifically to other monoclonal antibodies or other therapies like the ones that we currently use in the clinic. So there have been trials of a variety of other monoclonal antibodies, and I've just kind of emphasized the ones that are looking promising as available therapies, but there a handful of other ones have been looked at in people with symptoms. So solanezumab, I mentioned the failed trial as a prevention. It was also studied in people with symptoms and didn't quite make the cut. It came close. And some of the other antibodies, gantenerumab, some of the other ones, again, just didn't quite meet the goal that they wanted to. And it may be the case that to really show a clinical effect, you have to have that more dramatic lowering of brain amyloid. So some of the other antibodies just didn't show as impressive of an amyloid lowering effect to translate into a clinical effect. So again, the ones I'm talking about have that more dramatic amyloid lowering and better evidence of clinical benefit. So there are some differences, again, in terms of the kind of magnitude of effect. And these antibodies are all very different than the standard therapies we've had for the number of years, which are the pill medications that are designed to kind of rebalance neurochemicals in the brain, like the chemicals that help promote acetylcholine by blocking its breakdown, so the cholinesterase inhibitors, and then the other drug called memantine, which works on the glutamate system. So those are pills to kind of restore more normal levels of the neurochemical. They don't directly target amyloid at all, in essence. So very different approach.
All right. Oh, this one is for me. Will this presentation be available in recording? It will be available. It'll be up on YouTube in the next couple weeks. If you can search for it, or if you like want it right away, you can email me. My email is senava@med.umich.edu, and I will put it right here in the chat too. So yeah, just reach out and I'll be happy to get you that link. And I think we are all set with questions. So thank you so much. You have a great presentation and thank you coming in. Yeah, thank you for your time today. That was a lot of great information, Judy.
Great. Thank you, everybody.
Yes.